presentation. Okay, I think we're good now, guys. Greg, you want to kick off the meeting with a kind of an overview of what we're doing?
Sure, Joe. Welcome, everybody, this evening, sunny Cleveland, Ohio. It's been 75 degrees today, and I'm loving it. What you're going to hear today and tonight is some really good and promising news about the progress that we've made since the end of the year of 24 and what we've done now. You're going to hear from our team members, Matt Hummer and Felicia Hosney, on key ingredients that we need to solve in order to move forward into large scale operations. And with that, I'm going to turn it over to Matt Hummer.
Okay, thank you so much, and thanks everybody for taking the time to hear our updates. Hopefully you could see the screen, but the agenda and the items what we intend to cover for today are really threefold. The first and foremost is provide an update on our supply chain and how we're working to navigate that by establishing really a robust and redundant supply of this graphene material that's core to our technology and critical to commercial scale production. We've made a lot of progress in that regard. Second, regulatory progress. Felicia will be, filling you in on the progress there, but essentially at a very high level, we've kicked off the EUA process by submitting what's called a pre-EUA application, and that's for point of care detection of Ebola in whole blood samples. And so that's a promising step. And then third, and but certainly not last, is our manufacturing timeline. And we'll go through what that timeline looks like. But again, a lot of that timeline is really critical for not only the regulatory process, but achieving this commercial scale supply chain validation step that I'll spend a lot of time providing you an update on and how we're doing that. But after we accomplish those major objectives and move forward with those processes, we'll be very well positioned for our EUA submission using our pilot production and the bench testing. So turning to the supply reviewed more than 50 different graphene products from different vendors, not only through paper technical data sheets, but we've also had many conversations with the manufacturers of those products to really screen them and evaluate them and to determine whether or not they'd work for our product. And through that process, we've narrowed down to about 4 materials that now we're working with and doing very robust and advanced material evaluations. And then turning to the second technical issue, we've talked about our formulations and how they, establish and maintain superior and extended shelf life. And a lot of that has to do with the additives and the properties of the formulations and how they apply to the substrates that we're depositing these formulations on. So really to ensure this robust adhesion to the sensor substrate, it's critical for not only the sensor fabrication process, but also proving out this extended shelf life. So what we've been able to accomplish over the last 3 months is conduct these accelerating aging studies, these evaluations of more than 20 different additive materials that we think could potentially provide this level of adhesion that we need for this extended shelf life. And we've identified a single product that's actually, after preliminary evaluation, is delivering adequate adhesion, and that's been proven not only in material testing, but preliminary testing of these really accelerating aging studies, and we call these stability studies, is how stable is the product under accelerated aging conditions. And how we're conducting that study is through an incubation process of the material. So we're exposing them to very prolonged exposure of not only high temperature, but high humidity. And so things are going really well there. So, the summary here is, from a mechanical perspective, we've solved this adhesion problem. Now we need to work with this single additive we've identified to start to integrate into our formulations, along with this secure and redundant supply of this graphene material. So I just wanted to talk a little bit about how we're going about evaluating the functionalized graphene material that we're seeking to source a redundant and robust supply of. There's really two really parameters or characteristics that we're looking into and investigating. The first and foremost is the chemical composition of that material. And really, graphene, as you know, it's an allotrope of carbon, and it consists of really carbon and oxygen. And for us, through all of our advanced development, we've pinpointed what's the level of oxygen and what's the level of precise functional groups that are needed from this material that make up its chemical composition. And we've established tolerances for each of these properties. And so what we're doing is we're using advanced analysis to really be able to establish what the chemical composition is and whether or not a particular vendor can deliver that chemical composition within our established tolerances. The second characteristic is structure, or structural quality of the material. And if you think about it just as dimension, right? You have a lateral size, which is its width, graphene particle, and then you have the number of layers, which is its height. So we're assuring that the structural or dimensional quality of that material is uniform. And that uniformity leads to the stability or uniformity of also the chemical composition. So between these two main components, the chemical composition and the structural quality, we're able to deliver very uniform rheological properties. And what rheological properties means is how the material is adequately dispersed in fluids or dispersions and ink formulations, and how those dispersions maintain a proper particle size, a charge of the particle inside the dispersion, and most importantly, the viscosity, which is a measure of its thickness. And all these parameters matter for how we print this material on our substrate. So those are the things that we're looking to evaluate, and really how we do that. There's a lot that goes into it. And this is a slide that was presented the last investor call, but essentially we've established a very robust device master record, which is essentially a database of all the parameters that we're tracking really closely as the materials come in, and we characterize those materials and how those materials advance through our production process. So I won't spend a lot of time on this because I did talk about it before, but essentially there's the first and foremost parameters of the raw materials, and then how those parameters align with a lot of what I talked about on the previous slide, and then how those materials disperse into our formulations, and then how those formulations can be printed or produced in our methods of production. And then the last box offered to the right is really how we control the functionalization of these fabricated semiconductors that we're making. And then, of course, all the characteristics under test that we track very, very closely. So together, we can really begin to know all the data points and associated tolerance, not only for the raw materials, but how to track them throughout our fabrication process, so that by the time we're done making devices, we have a very high level of confidence that these devices will be highly successful against the FDA requirements for proving safety and efficacy. So I'll turn it over to Felicia to quickly go through some of the regulatory path forward. Felicia?
Yep.
Before you start, Felicia, I just want, there's people in the audience saying, well, what about FDA? What about this? What about that? I just want everybody to understand that we started the process. We submitted documents on March 13th of this year. So we have started the FDA process for the Ebola versus the blood specimen. So, and Felicia's going to go into a little bit more on that, but I want to make it very clear the process is started with the FDA.
Thanks. Yeah, thanks, Craig. So for it's been some time since our last meeting. So at the first part of this slide is just a little bit of a refresher on why the switch to blood. As many of you remember, we did engage with FDA in a pre-EUA submission some time ago for saliva and got some really great feedback from agency. But one of the things that we learned is our technology is new and novel, and also for the detection of the filoviruses, our sample type was new and novel. So FDA gave us a lot of great feedback, but the lift was really high. So what we did is took a look at their feedback and pivoted a little bit in the pursuit of blood targets. And so what that did for us is help give us some assurances that we're on the right path and in an essence, de-risk that regulatory submission. So as you can see here in the first table is a comparison of the sample type. So we're thinking about the Ebola virus, right? Typically in blood, you have a higher viral load, which is saliva is lower or unknown. There's not a lot of studies about that. Or multi-sample types, right? They're versatile sample use. And so what we did too is take our learnings and pivoted to that pre-EUA. We may have discussed previously, but just to lean into a little bit of what Greg just shared, the EUA process with FDA starts with the pre-EUA. So that is an actual submission to FDA. It is part of the EUA process. It is a submission that they will review, receive feedback, and then you execute on that submission and resubmit with the information that they requested. So it's absolutely a part of the process and a required part of the process. So that was submitted on March 13th of this year, and it has been acknowledged by FDA on March 19th. And I know a lot of folks have heard in the news about the upset apple cart to FDA and a lot of the government bodies. I'd just like to share with the team that the lead reviewer that we were assigned with this particular PEUA was also the same lead reviewer that we previously had with the saliva test, who is also still with the agency. That's great news for us in terms of continuity in the discussions that we'd had with her previously. So she's familiar with the test, the technology. So we're really looking forward to her feedback. With this submission specifically, there were some differences in what we put in for the saliva, where we're really focusing on that higher viral load with blood. So that helps improve our sensitivity. And we're leveraging that established regulatory precedents, right? A lot of the tests that are currently authorized for Ebola virus, they're blood-based assays. So it's a sample type that FDA is familiar with. And in terms of a clinical usage, it's a sample collection process or protocols that clinics are familiar with. So that positions us for what we feel is a better success story for EUAs. It's a stronger alignment with FDA cleared comparators. So these are blood-based sample type pre-CR tests. And then it helps support the clinical uptake with minimal operational changes. So again, sample collection, a lot of the containment procedures that clinics are used to today. So, and it positions us as a credible and compliant option for emergency diagnostics. Again, same processes and procedures, clinically, collection methods. So it increases the likelihood that we'll have easy or more favorable adopters.
Felicia, I'm sorry to interrupt you. We've just got a couple,
Sure.
we got a couple of questions, and I knew we are gonna save the Q&A to the end, but but you're on the topic anyway, and these two questions just came in. Can you talk a little bit about how we're starting with blood, but that is a pathway towards saliva, and we absolutely have
Yeah.
not abandoned saliva at all. It's just a next step kind of thing.
Yep, no, absolutely not. So going back to our submission last year where we had saliva, right? It's a new sample type and new technology. So you're compounding those complexities, which really, I want to say complicates it so much as it creates some challenges from a regulatory perspective because you're really fighting the newness on two fronts. So with this pivot to blood, what it helps us do is de-risk or eliminate some of that uncertainty and potential challenges from a regulatory perspective. And what we're really focusing on in that aspect is the newness of the technology and taking the sample type off the table. And what we can do, right? So once we are successful with the blood submission, will allow us to pivot, and that then takes the new technology off the table. And then we're focusing on the sample types. And also, in essence, by having that target, we're also positioning ourselves in a way where we can compare almost to ourselves from a clinical evaluation. So it really works out in the long run and helps smooth those potential regulatory wrinkles.
And I might add that when you look at our target list, one of those targets is H5N1 or H7N9. Both of those are classified as a variant flu. And the possibility for us to work and do research on that variant with chickens, that would end up using, you guessed it, saliva for the test, which is what we started out with. So that's also a possibility. And this is what Felicia is trying to explain, that even though we started with saliva, it's just not the right specimen for Ebola, but it could be very quickly for a variant flu, which we already created primers for and had worked.
Okay, great. Thank you, Greg. Felicia, is that it, or are you wrapping up? Or?
Yep, no, I'm good.
Good, excellent. Thank you so much, Felicia.
Sure.
Yeah, and so turning to the timeline, so just one point to add before that is, also part of our objective with, working with blood too was to really demonstrate the technology's versatility in a lot of different sample types. Because if you think about the path ahead, obviously just on the EUA and then stepping through to some of these more demanding regulatory pathways like a 510K or de novo, and then eventually finding ourselves really at the highest hurdle, which is a PMA. All these different types of targets require different types of sample types, whether it be saliva, blood. But if we can demonstrate that our technology is highly versatile across all these sample types, we're really in the process demonstrating or enhancing the value of the technology and the platform along the way. And so for us, when we get to a certain point where, partnerships with, supply or commercialization is demanding, we can rely on distributors to really drive the test in the market across all these sample types. But turning to the timeline real quick, so I'll just walk you through the key components of it. First and foremost, I want to start with validation of the reader and the cartridge production. On the last call, I think we were very clear to communicate that we've been working with our contract manufacturer over a period of time, and we've purchased all the equipment on the manufacturing line. We've validated nearly all of that equipment, and we've done production runs to prove that that equipment and the processes that we have in place are adequately manufacturing and assembling the cartridge and reader as intended and as designed. So for the most part, the point here is that The validation of the reader and the cartridge production has been established, and we've successfully transferred it from R&D to manufacturing. The only challenge really remaining is the scaled fabrication of the semiconductor. And so that's first and foremost. Felicia just spent a bit of time talking about how we did make this pre-EUA submission for Ebola in blood. That was done on March 15th, and FDA acknowledged it a couple days later on the 19th. By the end of this year, we're hoping to be able to make some kits available for research collaborators under either a LDT designation, which is a lab developed test, or a research use only designation, in which these devices can really be used by our collaborators to further validate the technology, gather data, and really help us advance manufacturing consistency in the science of how the technology is actually used with clinical specimens. Next is that we've made the pre-submission for EUA to the FDA, and now we have to await their feedback. I know Felicia mentioned that, but typically that can take some time. I think with the saliva submission, it took almost an entire year. So depending on when we receive that pre-EUA feedback, once we do, we'll be able to finalize our testing plans for validating the performance and efficacy of the product. And you know, we last investor call, we presented or spent a bit of time presenting all of our feasibility data. So we had to generate a lot of that feasibility data prior to submitting for the PEUA. And so that was a necessary and critical step. Depending on when we get that feedback from the FDA on our EUA plan, once we finalize it, we will quickly execute it and then make the submission for the EUA of detection of Ebola in blood. And so we will conduct that bench testing. We're hoping that, you know, the bench testing is very minimal, because that's the whole point of the EUA, to pursue a pathway where the burden of proof is a lot lower than conducting, say, full clinical studies. But after we get that initial step underneath our belt there, we'll be very well positioned to move into the more demanding regulatory pathways for hepatitis C, which is in blood, and then also the respiratory targets, which are in saliva. And between those two products and clinical testings, along with Strep A, we will be in a very good position to have FDA clearance for our suite of offerings, respiratory, strep, and hepatitis C, to be able to offer to doctors' offices and clinics that are right now either using one of two things: sending samples out to a lab like LabCorp or Quest and sharing revenue, or they're relying on inadequate or not so superior lateral flow antigen technologies. And we think at this point in time, we'll be very well positioned to expand partnerships for new test development, working with OEMs and very well-known medical distributors in the space to really get our technology to the market quickly. So this is the last slide, and it's essentially just a summary of what I said with you know, the cartridge that we're looking to commercialize, the very specific regulatory pathway we would be pursuing, and the target date. And again, we're focused on delivering filovirus, which is Ebola, and H5N1 for research use only. We think there's adequate opportunity for us to work with collaborators to not only validate the technology, but also establish some sort of revenue traction as well. And then that will well position us for the filovirus Ebola EUA somewhere around the second quarter, first quarter of 2006, depending on when we get that feedback. And then moving into the clinical testing for hepatitis C, which is a lot less demanding than the clinical testing for the respiratory, because keep in mind, for respiratory, we're testing for really 3 targets, would be SARS-CoV-2, influenza, and RSV. And then last but certainly not least, proving out the technology in a bacterial target, Strep A. So that's really what we're focused on. Of course, there's other opportunities for us given the nature of our platform technology, which we hope to share with you in the future. But we also want to be very clear that we're really focused on addressing the technical issue remaining and then executing on the Ebola EUA. Thank you.
Thanks, Matthew. And of course, we have Judy Mansour. Judy's Chief Equity Officer. And Judy, why don't you give us a little?
Sure. So good evening, everybody. Thanks for joining us. As most of you know, or everybody knows, we've raised a little over $20 million to date. We've been able to make all of this progress, as you know, that we've gone over this evening. One of the things that we wanted to do tonight was, as I've spoken with each of you, many of you asked, what we've been doing and where the money has been spent and so forth. And I've talked to you at great length about, our quality assurance process. So that's why Matt wanted to kind of raise the curtain on what that has entailed to get us to this point with the submission. So that's basically what we've been working on. I continue to talk with a number of larger institutional financial partners, and we continue those discussions. They continue to be interested. Those are longer ramp-up discussions than with each of you as individual investors. We are still accepting individual investments for the Series A. So if you remain interested in that, I welcome your calls. I will be in touch with everybody. And if you have questions, of course, don't ever hesitate to reach out to me. I'm happy to answer your questions. And if I can't answer, get Matt or Greg on the line. So that's really all of the update I have from the investment corner, but we still do have a Series A offering. So if you're interested in making an additional investment at this point, now that we are, closer to our milestones, be happy to talk with you.
Thanks, Judy.
Thank you, Judy. I'd just like to give you a quick financial overview of what we're doing. Our latest, one of our latest contributions was made in mid-January, and that was $3 million. And with that, we paid off most of all of our debt. That we have some due to the parent company, but we have about 2.3 million in assets, and that we own all the equipment on the line at our manufacturer and all the equipment in the lab. We have an additional $600,000 on deposit with our manufacturer. That includes cash and actual readers that are yet to be manufactured, because we have 7,700 already boxed that are paid for. And then also not only readers, but the cartridge material to make up to 13,000 cartridges, I think. So our burn rate's about 110, 115,000 a month. We're managing that very well. And that seems to go down a little bit every month. And we're looking to finish off that Series A raise as we talked, as Judy spoke of. And just as a reminder, that's a preferred stock, and it has a 2-for-1 payout versus on any kind of liquidation of any kind. So that's a good deal. And we're hoping to raise another, 7 million now, Judy? And that'll take us all the way through commercialization and scale up. And after that, we'll probably be very close to getting an FDA agreement on the Ebola by blood specimen, which would be very, very nice to have at the point in time. After that, we plan on raising about another 26 million for a large scale up of our hit list of what you see in front of you.
And, with regard to our Ebola submission, we have a new video on our website if you'd like to have a look at it. If you have friends that are interested in, our Ebola submission as a humanitarian effort, we have a great way to help us promote that.
And for those of you that don't know Judy, you should get to know her.
That's right.
Give her a call.
She's a very nice person.
Yeah, it's very nice.
Hey, Greg, as a quick question, somebody asked about the Series A and why it's still available. What is the plan to kind of close that out and protect the current Series A investors?
Well, like we said, there's 7 million left at $4.50 a share. And, we're talking to people now that are interested in executing a deal, several people. So when that's closed, we'll go to the next Series B, which will be to raise the 26.5 million. So it's a good deal. It's a good time to go for it because we're making really good progress. Like you heard tonight, we're finalizing on the supply chain and finding which company will produce graphene for us, the raw material graphene, in a highly consistent manner, so that we can scale up to build, millions and billions of cartridges someday. By the way, we might just like we mastered the ink in-house and now make our own ink. It might be that in the near future we may also bring in-house to functionalize what we call like raw graphene and functionalize it to our specifications. There's some equipment that are due, maybe about $100,000 worth of equipment that we'd have to buy, but that would make us completely independent of everything. We would be manufacturing all of our own ink, functionalizing all of our own graphene, and that would be something.
Great, thank you, Greg. Let's get to some of the Q&As here that we haven't we've touched on a few of these, so that's good. One quick question, and Matt, I know you and I talked about this recently. What are we doing to, or what would we do to protect the IP from competitors once we go to FDA? Well, as we cycle through FDA.
Yeah,
Ron,
that's a great question. So we do have lots of patents in this space. We have 26. If you look at our patent portfolio, we've done a really good job of leveraging really foundational IP that was initially examined and awarded in 2010 time period. And then we found a way to build a portfolio of about 26 granted patents, and then we probably have between 8 and 10 pending, which are new patents and continuations from granted patents. So there's kind of a couple things. One, we have really good IP that we can continue to expand and broaden. So that should help us from actually a legal perspective. The other thing is we're moving the most important patents that we think into prosecution internationally. So in the EU and some of these other countries, so that we have patent protection outside the US, which is a good step, but it's also expensive. So you need to be selective on which patents you actually do that. Third, a lot of what we're doing, there's a lot of know-how involved, obviously. So, there's a lot of things that we've just learned. And just to be very straightforward and transparent about it is, I would think it would be incredibly hard to copy or duplicate what we're doing, even if you knew the exact ingredients and everything, because it's a complicated process. And it's more so about the R&D and the things that we know that make our IP and our know-how together a very powerful path for commercialization. Another thing that I also do want to kind of mention that it is really providing some level of challenge to us in this space is, graphene sensors and other biosensors or other types of sensors, they are extremely versatile, and we tout that as an advantage of our technology, and it's true. You can functionalize a sensor to detect just about anything, And I think that's a good thing, but it's also a bad thing because, when we're starting to work with different vendors or people in the space, even the manufacturers of the material, they make the material, but they're beginning to evaluate and understand and work in adjacent markets. And we've learned that there's a good amount of conflict of interest when working with a particular vendor around, our core requirements. And so, sharing those core requirements and ensuring that a lot of this confidential information is protected is particularly, we pay a lot of attention to that. But having said that, it makes establishing these partnerships with some of these vendors challenging. There's a lot of effort in legal documentation and NDAs and agreements that we need to be very clear about. I think a lot of folks on this call that have been with us for this journey understand that, we've had our, experiences in this realm. And, as a result of it, I think doing what Greg suggested is the end all goal for our company really is to own the whole fabrication process, including the raw materials. Because if you think about what we're doing is we're using a raw material and we're formulating it and we're making it into something. But if we don't have full control over that raw material, that leaves us really exposed in terms of supply chain, in terms of the repeatability that we need to get from it. And, that's something that's a significant really issue for us. And so all this is to say is that, we are gradually trying to grow our capabilities to really cover the full gambit. And I think at that point in time, and we've done it across our journey, right? We started not making our own inks. We started not printing our own inks. We've learned how to do all these things, and it's taken us a bit of time, but we've done it. And in order to actually be successful in this regard, it's going to take that and then also perfecting it and working it. So um, hopefully that answers the question about
Yeah,
IP.
great. Thank you, Matt. Appreciate it. One other question, Greg, maybe you can touch on this and probably speculation, but what effects, if any, will the current administration in the White House have on FDA process?
I
Yeah,
think
go ahead, Matt.
well, I actually, well, Felicia's not with us, but I've kind of been providing a response to a lot of this, and I'd like to just piggyback on what Felicia already mentioned, which, the FDA reviewers in terms of medical device, like that's actually a revenue center for the government. The FDA process is expensive; it generates fees. There's lots of biotech companies that are approaching the FDA and trying to commercialize. So a lot of the FDA reviewers are intact. They're all still there. They're progressing as normal. A lot of the process for actually evaluating safety and efficacy of technologies is written into the congressional code, right? CFR 2021. So those things, agency heads don't have the ability to just change those things. It has to take pretty extensive legislation. In fact, the last time a lot of those things were changed was quite some time ago. So one things that are actually welcomed by this administration is that they might simplify the process, make it even less expensive. And I think the biotech community would be very welcoming of that. But for the most part, a lot of the turmoil that you're seeing within the federal government hasn't really affected the FDA, and particularly our reviewers, as Felicia.
Most of people's fortune is spent in the last 90 days with cancer and other kinds of diseases, communicable diseases. And we can shorten that. We can decrease the numbers of people that by quickly diagnosing and treating. And that has a definite, ring of savings to it, which this administration is quite frankly interested in. I mean, it's nice to be able to have a product that where all the vectors are in line. You've got low cost, high production, and quickness of speed to treat. That's hard to beat today.
Yeah. Thanks, Greg. Matt, quick question. Looking ahead past EUA and into the 501, 510K de novo approvals, do you foresee seeking additional pathogen targets in parallel?
Yeah, I see that question. Certainly. I mean, and I think that's kind of really what we intend to do, because the ability for our technology to be manufactured pretty quickly by just depositing a different biomolecule on the surface and not have to redo all the chemistry as part of the chemical based assay like PCR or a digital process. So yes, certainly. I think, once we establish the EUA, we'll be well positioned to pursue partnerships and commercialization pathways with the right resources to really execute, trials simultaneously. And, we have a very limited scope for what we're focused on right now. But once we validate that we can, fabric or address the supply chain issue and then make a lot of these, units at scale, we'll be very well positioned to pursue lots of things.
In
tandem.
fact, on that hit list that Matt showed, there's only one item on there, that's strep, that we haven't produced sensors for that have worked. So, and these are key diseases in states that can have lots of savings, diagnosed early and treated early. And, it's just a matter of time. Now, what I want everybody on this call to understand is when we go to do clinical trials on everything else but the Ebola, because it's a EUA, those clinical trials cost money. And that's where most of our money in the future will go, because we've already know and formalized many of those targets on the target list already. It's just a matter of scaling up. But like, for instance, on hepatitis C is probably going to take $800,000 to $900,000 just to do the clinical trial. And the clinical trials for respiratory, back when we were going to do them, was almost $6 million. So, most of that 26 million that we want to raise after the Series A will go to earmark to clinical trials, like Matt said, to do a lot of these simultaneous.
Great. Thanks, Greg. I think that wraps it up for the questions. We've touched on most of these topics that have come through. Greg, do you want to give
Joe,
any final words?
I have just one more.
Yeah.
There was one more about. Oh yeah, no, the graphene requirements. And yeah, somebody asked about the graphene requirements, whether we have them established. Yeah, we do. We have a very detailed, sheet of all of our requirements that outlines, the particle size of the material, how it's made, its zeta potential, which is its electrical charge in a solution. What's the carbon content versus oxygen content, the very specific functional groups on the material, and the tolerances for all these parameters. We've evaluated how these things change in the raw material and then also through our fabrication process, so we have a good handle on that. And yeah, we've shared a lot of these requirements with these very few vendors that we've down selected to, and for some of them, they're helping us modify their materials towards those requirements. And for others that don't have that capability to modify their materials or very specific requirements, we're working with other vendors' capability to do that ourselves. Because if you think about it, if we can move along through just buying off the shelf graphene, and all it has to be is consistent and uniform, and that exists out there. And we developed this functionalization process, which is really transforming the off-the-shelf material to our customer requirements. That's really, what we're focused on. But again, in order for us to make progress, we don't have to do that. But eventually, I just wanted to mention.
Okay, that's great. Thank you very much. All right, I think we are good. Greg, any final thoughts?
Just thank you all for will succeed at this. This is technology that is just wildly amazing when we talk with people that are in the know. They just taking a superconductor like graphene and turning it into a molecule. It's just so elegant. So thank you all for standing by us, and we hope to continue
Hi, yeah, I think we're